Hi guys, welcome back to the channel. Today I'm going to talk about a particular study on a newly discovered antibody against the SARS-CoV-2 virus. If you are new to the channel, I'm Dr. Han. It's lovely to have you here. In this channel, you will find science review content and updates on the latest global health topic. I also like to share tips and tricks for students' academic and personal development. If these are your interested topic, please consider subscribe to the channel. If you have already subscribed, thank you very much for coming back. So during early September, the University of Pittsburgh School of Medicine published an article talking about they discovered a new antibody that can effectively against the SARS-CoV-2 virus. Now this topic is very exciting to me because it's somewhat related to what I do for my own research. So today let's take a quick look on what this study is about. So today we are going to talk about this new tiny antibody discovered by the University of Pittsburgh, and let's look at it briefly of what it did. So a little bit of the background. Earlier this month in September 2020, University of Pittsburgh School of Medicine published a paper describing finding out a new tiny antibody that can effectively against the SARS-CoV-2 virus. And it showed ability to neutralize the virus both in the petri dish experiment and as well as in animal models. And let's look at what they did, okay? So before we can talk about what the researchers did in the experiment or in their study, let's first have a basic understanding of the structure of antibody. So here you are looking at a classical shape that people draw, which is a Y-shaped and consider with two parts, basically a center portion and a side portion. Now the tip of this fork-like structure is usually where they bind to antigens. We call it antigen binding regions. If you break down the component of the antibody, you will find it becoming two parts. The center part we call it heavy chain, which is the bigger part, and the side small chain we call it light chain. Now you can break it down even further and manipulate it in different ways for different purposes. Here are some of the examples. Now this is not a complete list, they are just some brief examples. If you're just looking at the orange and red part, we can call those parts variable fragments, okay? These regions, because they bind to antigens, so they are actually different for a different type of antigen, so they are highly variable in a sense. Now the researcher can also put a linker between the two fragments of the variable regions, and we call these entities single chain variable fragments. And lastly but not least, just a small tip, okay, this red orange tip from the heavy chain, we call it heavy chain variable fragment. It can also participate in binding of antigen. Some literature called this body nanobody, or abbreviated as VH. Now this body is actually important in today's talk because it is what the University of Pittsburgh found and published. So how do they find this small antibody to do binding on SARS-CoV-2? They utilize a technology called Phage Display Technology. Now this technology has been around for 3 decades already, and it has been shown to be successfully finding different drugs and get those drugs to be approved in the market. Now the drug that I'm referring to at this point is called antibody drugs. Okay, now to begin with this phage display technology, first we need to understand what are phages. Phage are actually viruses that specifically attack bacteria. They are harmless to mammals and humans. And in Phage Display Technology, researchers infuse human DNA into the phage particle where it can express a small piece of amino acid and or a type of a human antibody fragment on their surface. In our case, the researcher put in the VH fragment gene into the phage and therefore displaying the VH fragment on the surface. And usually these phage display technology are displayed into a phage library here on the right hand side. That means the researcher have millions and millions of different variable fragments are being displayed in this big library, and the experiment goal is to find out one winning ticket or several winning tickets that can bind to a specific target, and in this case it is the receptor binding domain on the spike protein of the SARS-CoV-2. So in this whole experiment, basically the receptor binding protein is fixed on a flat surface, we call it a pan, and during this panning experiment, the phage library is being incubated with this antigen and through a repeated cycle, and eventually only those that bind to the receptor binding domain very well and very specific will be selected for further investigations and amplifications in the bacteria to make more and more of this piece of specific binding elements. Now, going to the result, after this type of experiment, the researcher screened out a VHantibody or this tiny antibody, they name it ab8. Now, they further manipulated this fragment into a bivalent VH molecule. Basically, they put two of those together, link it in the middle, and also fuse with the constant region of an antibody. So they make it a little bit bigger than what it was originally discovered. And this newly discovered bivalent VHantibody can neutralize SARS-CoV-2 in vitro, i.e. in cell cultures, and as well as neutralizing SARS-CoV-2 in mice and hamster models. So if this antibody is so good, how can it become a drug for us to use to treat the disease? Now, first of all, we have to understand this experiment only did into animals, and animals have a smaller circulations, and the effect that we can observe in animals may not be as well translating to the human study. So scaling up to human circulations can be very difficult. And there are several questions we need to address when we are dealing with antibody-drug or any other drugs. The first question is, how does the antibody affect the human body? We also call these questions pharmacodynamics questions. Another set of the question is, how does the human body affect the antibody, aka pharmacokinetics? So the biggest question is, how long will this antibody drug stay into our body? If it is very quickly being eliminated, it will not do its job and it would be very much useless. Now, we can do some modifications to enhance the antibody profile and make it stay longer in our body. However, this may also induce immune response. That means the body generating immune response and attacking the particular drug and makes the drug useless. So to learn more about this topic, I've included the original link to the article in the description box down below. Now in the past, actually in April, I also developed a full-length lecture on the concepts of phage display technology and ligand evolutions. And if you are interested in learning the whole concept, please feel free to check out this video in the description box below and up at the corner. Now, I have to warn everyone, this is a full-length lecture intended for pharmacy students, so there are a lot of technical terms. So I hope this video gives everyone an idea of how antibodies are being discovered outside of our own body. Alright, that is all for this week's COVID-19 update, and I'll see you again next week, Sunday. Bye.